Okay, can you all see my screen?
Yes.
Okay.
We actually see your entire screen. Maybe you can, oh, is this a PDF slide maybe?
No, it's through slides. Let me see if I can try this. Okay, do you only see my screen?
We see your browser as well, but I guess it'll work.
Okay. Wait, sorry. Do you see my speaker notes?
That we do not.
Okay, just making sure. Okay, sorry for the delay. So my name is Lisa Martinez, but I go by Lizzie, and my title is Comparison of Hyperosmolar Volume Reduction between Astrocytes and Neurons in a Living Brain Slice. So to give some background, I will be focusing more on epilepsy on a cellular level. So as we all know, or you may not know, epilepsy affects like over 2.4 million people. And while there are anti-epileptic drugs, it doesn't work on everyone. To be precise, 33 of these cases are unresponsive to these drugs. And because of this, it is imperative to investigate on the mechanism of epilepsy and what the lab I'm currently in, which is Todd Fiocco's, which we're trying to study, is cellular swelling. So some of you may be wondering, oh, why are we studying swelling? Well, epilepsy is categorized by seizures, and the seizures result from a swift increase in volume within the cells, and this can lead the tissues to swell and reduce the brain's extracellular space. And because the swelling can cause the cells to be in close proximity, as you can see on the image, they're all bumping into each other, which again increases neuronal excitability, which again leads to seizures. To give an introduction on like the actual technicalities on this. So first we have to define astrocytes. So astrocytes are a type of glial cells that are responsible for maintaining extracellular concentrations, and they also shrink during cell swelling. On the other hand, we have neurons, which are the basic units of cells that send and receive signals in the brain. Previous literature has also indicated that neurons aren't as osmoresistant, which has been clearly observed. However, we haven't answered the question why they shrink, because contrarily to popular literature, the lab has actually found our neurons can actually shrink. And so this is important because previous literature said they can't shrink and they were operating on the assumption. So that means we have to kind of redo all of this knowledge that we had and calibrate the techniques that we had before. So the purpose of this experiment is to research volume reduction for astrocytes and neurons while determining their specific contributions to epilepsy and neuronal excitability. And this leads to our hypothesis. So our hypothesis is if a hyperosmolar solution, ACSF, is applied, then there will be a greater amount of shrinking in astrocytes compared to neurons. So to explain the reasoning, let's define hyperosmolar. So it refers to the increased osmolarity of the extracellular ion concentration outside the cell versus inside. So this basically means that it induces swelling as a cell would want to maintain balance between both of the concentrations. And how do we increase the osmolarity without changing the extracellular ion concentration? Well, we found out it was actually mannitol. So in a previous study by the lab, mannitol, as you can see in the figure C, mannitol ended up shrinking astrocytes significantly more than neurons, and the control ACF, which is shown on the right, another word you could find it is baseline. It shows that it returned to normal after exactly 5 minutes after you apply the solution. So if you can see in the middle image, you can see the green color that indicates the reduction of the soma. And as you can see, the neurons shrank less in astrocytes. And this is reasonable to assume and actually see is because neurons have a rigid cytoskeleton, so it's harder for them to shrink versus astrocytes. You can think of it as trying to mold hardened clay. And in this study, this also shows that neurons are capable of shrinking when osmolarity is altered, which again is contrary to popular literature. And for this experiment, I would just be trying to replicate the same results, but with sucrose, which has the same ability to shrink without altering the concentration, as we do want to maintain the artificial cerebral spinal fluid. as we're not working with cell cultures, we're working just with the cells itself. So it's harder to maintain that exact condition or environment. And the results I expect to see is that it would replicate whatever we had with mannitol. So to go into a little bit of the methodology, it's in 3 steps, which is slice prepping, dissection, and volume imaging. So in terms of like slice prep, what we do is we get the mouse, we dissect it. We only use the hippocampus of the mouse. And in terms of like the actual procedure, we get like ice buckets filled with slushy. So we do want to preserve its health while it's cutting, because once it's dead, you want to swiftly preserve the health. Otherwise, you won't be able to get enough volume imaging from it. And then after that, we have to bisect the slices of the brain itself. And then we transfer it to a vibratome, which can be seen here. So a vibratome is that machine on the far left. On the top, that's where the bisection happens. And on their bottom of that first image, that is where we actually see the cells. So in terms of like, how do we actually see, the astrocytes and neurons? So astrocytes are marked by SR101, which is basically just a green fluorescence. On the other hand, neurons are marked by a green fluorescence, which is EGFP. And in terms of the actual solution protocol, what we do is relocate the pyramidal layer, which is shown on the left, and we proceed to find like a well-defined neuron on astrocyte, and I'll get more into detail for that in the next slide. So just to explain what's going on behind the scenes, on the right, within 8 to 10 minutes, an image is taken as they are washed by ACSF. We wash it just because we want to see, like when you wash it, you just want to see how it impacts it, and regular ACSF, and then we do mannitol, and then we repeat that process. In terms of why we use these specific like minutes, it's been a standard protocol for the lab, as this was, the lab was founded. So that's why this procedure has been modified to look like this. Right now, to explain like what like exact images are imaged, we go here. So on the left, you can see how this is ideally how you want to like image. However, on the second image within that same one, you can see the ROI, which is the region of interest, which is the circle. So you want to make sure that there isn't any fleckling, that it's clear, it's distinct, it has well-defined borders. Those are the main criteria for imaging. Otherwise, you're going to have too much noise, as in you're not able to capture the astrocyte or neuron. And in terms of how the imaging process works, once you find that astrocyte or neuron you want to image, you want to basically each time you take a picture of it, you're collecting stacks of images. So think of like you're having like a 2D image, and then once you create stacks, you can actually see it like a 3D, just to help give some insight into that. And then once you do that, the software will help have a cleaner version without the background, and then you're going to get a binarized image, which basically just makes it easier for image recognition, and you could actually calculate the volume reduction through the software. And again, a second ROI will be drawn to quantify the volume, and that software will help you analyze it later on. And I guess, and for moving forward and final thoughts, I expect, as I said before, to have the same results as mannitol, and will closely resemble our previous data, and hopefully move forward into trying to find out like how do we now like stop the seizures and epilepsy. Another thing is like for technical aspects, just to perfect dissecting and imaging, because as you know, like your whole data relies on the health of the cell, since you're swiftly moving it from, from the mouse to like the vibratome and then to the actual microscope. So you want to make sure the cell is like in very good health. Otherwise, it's really hard to image it. And it's harder to get a pyramidal layer as well. Again, for further research is just to understand the mechanisms and why neuron shrinks, and how can we prevent that for going forward. And I just want to acknowledge the Marquis Star pre-training program, which got me into research, and Rebecca Brown, my PI, Todd Fiacco, and the grad students who have helped train me, which is Erin Welch, Alex Bilas, and Sandhya Surinam. And that's the end of my presentation.
Thanks, Lizzie. Questions from the audience? So I'm not really a biologist, but I'm interested in like when you said dissecting the mice. So do you do that, or do you assist the grad student? Like how does that process work?
Yeah, so I'm actually the one who does it. So what I get is we have like a glass bowl and it has isoflurane, which is very toxic to mice. So we get an actual live mouse and then just put it there. And when it's barely gasping for its life, it's about to be like 2 seconds before death, we immediately have to grab the mice and then put it onto like the platform and like, pinch its feet to make sure, it's not alive. We don't want to cause like animal cruelty. And then you just immediately like behead like the head of the mice, and then you immediately have to start like cutting around and like getting rid of all the fur, the skull, and then until you get into like the brain, that's when immediately transfer to an ice bucket that already simulates the environment that the brain is, which is cerebral spinal fluid, and then you want to bisect it, and then once you bisect it, you put it into the vibratome.
Wow. So do you get practice? How much practice do you get? Or you need to be pretty good with your hands, right?
Yeah, I'm still getting practice in it. I believe I got like 10 times already.
Okay.
Yeah, it's just I think the hard part is actually like when you're cutting, because it's so small, it's easy to puncture the brain and you can actually damage, the slices of it later on. So that's like the hard part. You want to make sure you don't puncture the brain while you're dissecting.
I see. And you do the imaging too?
Yes.
Wow. Very good. All right. So
Um,
Michael, I think in your hand.
I just had a question. So the goal of this was to see if treating brain cells with this, what was it, mannitol, was going to shrink the neurons, and that's meant to potentially alleviate epilepsy, or like maybe not directly, not as directly in that sense, but if cell swelling, neuron swelling is having a role in epilepsy, then something that could alleviate neuron swelling would potentially treat it. Am I getting that right?
I not exactly, but you're close. It's basically I'm using sucrose to see if the data matches with mannitol, and if it does matches, it can help us learn how to stop that swelling using that same solution. And yeah, the goal would be to stop epilepsy.
Cool. So that brings us to the end of this.